All right, in this video, we'll be talking about transcription RNA processing, more so on the latter side, RNA processing, talking about the modifications that are made to the RNA after transcription, making it into a full-blown messenger RNA. Real quick thing here to talk about the differences between prokaryotic and eukaryotic transcription. Remember, in prokaryotes, there's a single circular chromosome obviously there's plasmid DNA inside the bacteria as well. But in eukaryotes, you have multiple linear chromosomes. And so, and also the bacteria, the prokaryotic cell doesn't have a nucleus. And so the ribosomes and the DNA are in close proximity to one another, whereas in the eukaryotic cell, in order for the messenger RNA to be translated, it has to leave the nucleus and go and find a ribosome. And so, to that end, in prokaryotic cells, many times the messenger RNA is being translated even while it's still being transcribed. And so, it's happening very, very quickly, as opposed to the eukaryotic cell, which has to be transcribed and then has to leave the cell and then has to go through the process of translation. It would be akin to, I use the illustration of me copying out of a textbook for transcription. Imagine me copying out of a textbook, and while I was doing so, multiple people were standing by and translating each word as I wrote it at the same time. That's what's happening in a bacteria, as opposed to me transcribing a portion of a textbook and then sending that to another room and then that being translated separately. And so that's kind of the difference between prokaryotic and eukaryotic transcription, translation, protein synthesis in general. So let's talk about the modifications that are going to occur to the RNA after transcription in a eukaryotic cell. This is not happening in a prokaryotic cell because, well, again, they're happening at the same time. And so a eukaryotic cell has to be modified, and largely because of the difference between the environments in the nucleus and outside in the cytoplasm. Cytoplasm is a dangerous place. And so, so the messenger RNA, which is what you see here, this protein-coded sequence, is going to be protected by several things. And the first thing is going to be called a poly-A tail. It's called a poly-A tail because there are 100 to 200 additional adenine nucleotides. This thing says 50 to 250, and so it's in there somewhere. And basically, it's just a long string of adenines. And what this does is it increases the overall stability of the RNA molecule. It prevents enzymatic degradation as well. And so once that messenger RNA goes into the cytoplasm, enzymes are going to be breaking it down immediately because the cytoplasm doesn't need a bunch of old messages floating around. And so it's constantly breaking those things down. Those nucleotides are returned to the nucleus in order to do some more work, so no pieces are wasted. But that messenger RNA has a job to do before it's completely broken down. And so this poly-A tail gives those enzymes something to chew on before it gets to the important parts. It's kind of the way that I think about it. All right. There's also a GTP cap that is going to be added. It's very similar to the poly-A tail in that it provides a protective sort of mechanism. And when I say GTP, think ATP, except for with it, and not an adenine, but a guanine. And then you have that 3-phosphate sort of thing going on there. Not only does it protect, but it also helps the ribosome attach to the messenger RNA. It's kind of like a point for them to connect. That GTP cap kind of signals the ribosome to come and start translating this particular section of messenger RNA and gives it a place to kind of attach and get started. So that is 2 modifications, and the third modification is basically going to involve these 2 things called introns and exons. And so what are introns and exons? I'll go back to my textbook illustration. So let's say I have a textbook and it's 1,000 pages long. 800 of those pages are blank. So a large portion of the textbook is blank, and it's not just one section in the back. It is scattered throughout the whole thing, and all throughout there are sections of reading that you need to know, but you have to skip through a bunch of blank pages in order to get to that. That's kind of like our DNA. It has these sections of actual material that's necessary and then a bunch of blank pages. The introns are basically like the blank pages. The exons are the actual genetic sequences that need to be used in order to make proteins. And so, in order for a messenger RNA to be made into a protein, those introns or those blank pages have to be removed, and those exons have to be connected together. And so, the third modification that happens is the introns are going to be removed or excised, is what probably more technical term, and those exons or introns are going to be excised, and the exons are going to be connected. Now, one interesting thing that can occur with this is something called alternative splicing. So this picture shows really well the DNA section up there. You have those exons again. Those are good material. That's actual DNA that's necessary for the creation of a protein. And this particular is showing you that these 5 exons make up a protein. And so you see all the blank sections in between. Well, then the RNA is transcribed and you still have those blank sections. So the introns, which are represented by these non-colored portions, are all taken out. And this is what you'd expect to see, right? 1, 2, 3, 4, 5. What alternative splicing is essentially putting those exons in a different order. Now, what is this going to do? What we've learned early on when we talked about proteins, that proteins have this sort of structure to them, and that primary structure is just a string of amino acids, which is what this is going to code for, right? Well, 1, 2, 3, 4, 5 is going to make a different string of amino acids than 1, 2, 4, 5 is, and then 1, 2, 3, 5 is going to make. And if you have a different string of amino acids, when that protein folds up, you're going to have a different shape. And if you have a different shaped protein, what are you going to have? You're going to have a different function. And so essentially what goes on here is you have one gene, but this one gene can be responsible for coding for several different proteins in kind of what would be called like a protein family. They all come from the same genetic material, but they may have differing functions, slightly different. Maybe they all work together. There's a lot of research on this because we actually have a lot more different proteins in our cells than we do genes. And this is one of the reasons why that occurs.